Are there sex differences in Alzheimer's disease? If so, how can we find them? Well, gene regulation is complex. At a basic level, transcription factors come together to form complexes. These complexes bind to DNA, recruit RNA polymerase, which then transcribes a gene. Some genes also code transcription factors, which then form complexes of their own and regulate other genes. In many ways, cells are like little computers running transcriptional programs that will determine the state of the cell. Sometimes something goes wrong in the cell's programming, but how can we learn and debug the transcriptional programs in a cell? Well, let's go back to school. At school, some teachers may do a poor job communicating with students, resulting in poor student performance, while others may be excellent teachers, with all students passing their exams. On the other hand, a teacher may be teaching effectively, but some students may not be paying attention. In message passing terminology, we can say that the teacher is responsible for teaching, but the students must also be available to learn. Transcriptional programming is similar, with transcription factors responsible for regulating their target genes, resulting in more or less gene expression. And that's where PANDA comes in. PANDA uses message passing to estimate the connections between transcription factors and genes. Once we know something about these connections, we can begin to figure out if errors in the transcriptional programs associated with Alzheimer's disease are different in men and women. Starting with gene expression data, we estimated transcriptional networks for men and women, both with and without Alzheimer's. We then looked for changes and found something interesting. Although energy metabolism genes are expressed at higher levels in normal females compared to males, there were no sex differences in Alzheimer's disease. However, in the transcriptional programs, we saw increased targeting of these genes in females and a decrease in males. When we looked at what might be driving these changes, we found a number of transcription factors that respond to hormones that appear to be most responsible for the differences. It's exactly what we might have expected to find, but we couldn't have without knowing the networks driving the transcriptional programs of these cells.